Okay, so bacteriophages, these are viruses that only affect bacteria. They have this very kind of alien looking structure that we saw before, the isohedral head with the tail. And we'll talk about how bacteriophages go through those four steps from attachment to assembly and release. So attachment with the bacteriophages, these tails will attach to the viral receptors on the surface of the host cell. And this is a very specific ligand receptor bonding, like we've talked about in the previous semester, right? So that the there's a special configuration on both the receptor and the tail of the bacteriophage, where it's kind of a lock and key type specificity. So any given phage can only attach to certain receptors, and this really is going to limit the cell types, or the types of bacteria that it's able to affect. So this is the first step. Once it's attached, we have injection of the viral chromosomes. So the chromosomes up here, here we have this T4 phage. It has DNA. Um, in the head, it actually is injected kind of like a syringe through the cell wall, the cell membrane of the bacteria. And then we have the phage DNA is inside of the cell itself. Now, those next steps, so 3 and 4, how they go about is dependent on the bacteriophage type. And we'll talk about two major cycles or ways to kind of act once the bacteriophage genome has entered a bacterial cell. And so first we'll cover virulent bacteriophages. And so the T4 that we've talked about before, this is a type of virulent bacteriophage. After it enters after the DNA enters the host cell, it then enters the lytic cycle, where it's going to reproduce, make lots of new viruses, and then kind of burst out and kill the host cell. And the other is a temperate bacteriophage. Our example here is lambda. In this case, after entry, the temperate bacteriophage may enter the lytic cell, right? Or sorry, the lytic cycle, where it'll make lots of new viruses and kill off the host. But it may instead enter this lysogenic cycle. And the lysogenic cycle is a kind of a latent state where the chromosomes are just kind of, or the genome is just kind of hanging out within the bacterial genome. And it can switch if it does first enter the lysogenic cycle, it can switch the lytic cycle. So let's look at this virulent bacteriophage first. So with the lytic cycle, we have the viral attachment, just like we've already talked about, viral entry, so injection of the phage DNA. And then one of the kind of first genes that will be just transcribed and translated from this host DNA is going to be genes for enzymes that will then digest the host DNA. So basically just kind of cut it up. Now the host cell, its machinery is just going to focus on replicating the virus and not transcribe its own genes. And so that's what happens next. Replication, so production of more chromosomes, and the capsid protein, so the parts that are going to make it up, they're going to self-assemble inside of the host cell. And then the virus later on. Some of the genes that are expressed will be for a lysosome, which basically is going to cut up the bacterial wall. And most often, we're going to then have water fluids can go into the bacterial wall and can cause the whole cell to burst. Either way, the bacterial cell wall is going to be kind of cut up and compromised, and these viruses are going to be released. Okay, so this is the Lytic cycle, and this happens quite quickly, and it's producing, it can produce thousands of new viruses in, a short amount of time. 20, we watched the video in class where it was just 25 minutes after kind of the viral attachment after the first infection that the cells were lysing out the new viruses. All right, so the temperate bacteriophages, they can go into the Lytic cycle, but they have this kind of other option where sometimes they enter this lysogenic cycle. And in here, the viral genome is going to join the host genome, so kind of become part of the bacterial chromosomes. And it's silent, and by silent is in quotes here because it may have some effects on the cell. And then it's going to be replicated with the host genome. This is cut off just a little bit, but this just says host here. So these steps, same as before, it's going to attach and inject. But instead of any replication going on, that phage DNA is going to join and integrate into the bacterial chromosome. So you can see this little bit of blue that's just joined in with the red bacterial chromosome. And when this happens, that is referred to as a prophage. So this blue part that's just kind of hanging out, and it's not replicating, it's not having any kind of major effects in killing off the cell at this point, is referred to as the prophage. So then as the bacterial cell, goes through mitosis, and makes copies of its own bacterial chromosome and then, divides. Each time that happens, it's going to copy the prophage, the phage DNA, along with its own bacterial chromosome. And so all of the copies that it makes are infected with the virus, right? So here's one of the daughter cells with the prophage. And it's important to note that during this, at least for the most part, our phage genes are not being transcribed. So they're just kind of latent, hanging out here, and not having much of an effect. It's still replicating because each time the bacterial cell replicates, it makes another copy of itself. Okay, so with these temperate bacteriophages, they can do either, right? So they can, depending on basically the environmental conditions that the host bacterial cell is experiencing, you can either have the lytic cell being induced, where it's going to make a bunch of copies of itself and just kind of immediately destroy that host cell, or it can just integrate and replicate as the bacteria replicates. If it enters the lysogenic cycle, it can, switch as environmental conditions switch, and the lytic cycle will be induced, and it will replicate and produce a whole bunch of viruses. Okay, and so let's see, let's just compare that a little bit further, right? So lytic cycle, both types of phages can enter the lytic cycle, destroys the host DNA, produces a lot of new phages. And then the cell will lyse, and all these new viruses are released. Lysogenic, only the temperate phages enter the lysogenic cycle. The genome's integrated into the chromosome as a prophage. It's replicated, passed on to daughter cells, and depending on environmental conditions, can be induced to leave the chromosome. So the prophage will separate from the bacterial chromosome, and it can initiate that lytic cycle. Okay, so how how and why might I'm gonna pause this for just a moment. Okay, so how does it decide? So on the kind of molecular level, there's these two important um, proteins that are involved. One is referred to as C1 or the repressor. And so it's a transcriptional repressor. It's going to bind to promoters and keep off those early lytic cycle genes, right? So it's going to block kind of that lytic cascade, um, so that the the viral genes aren't going to be transcribed. They're not going to cut up the host DNA and start making copies of it themselves. Um, and then there's also this lambda C2 sensor, and this is a transcriptional activator. So it'll bind to DNA, and it's encoded in the lambda phase genome, and it's kind of a switch, acts as a switch in the lambda phage pathway in terms of which one it's going to, um, which cycle it's going to be in. Um, and so this is based on kind of environmental cellular conditions, um, as to whether it's going to, uh, make a bunch of copies and lyse the cell, or it's going to enter the lysogenic cycle. And so how does this work? So the C2 isn't very stable, and so they break down a lot and need to kind of constantly kind of be made. And so we can look at this one condition. So whether we have well-fed bacteria, so on the left here is the well-fed bacteria condition, on the right, the starved bacteria, right? So this is our host cell. Um, All right. And so in this condition, it's well fed, so there's lots of ATP available within the cell. In the starved bacteria, we have a lot more ADP. So we don't have the conversion to ATP happening as much. So the phage DNA in both cases will transcribe this C2, our kind of uh, switch protein. Um, when the bacteria is well fed, we have ATP activating the host protease, which is then going to break down the C2, right? And so when that happens, C2 isn't available, right? It ends up uh, being really important for C1 being expressed. So when in the starved bacteria, the inactive host protease are not affecting C2, so it's stabilized. And C2 continues to be produced, and it allows for the expression of C1. right? And so C1 is around, and that causes or that blocks the lytic phase. It blocks the phage DNA from expressing the genes necessary to initiate the lytic phase. So basically, if the bacteria is starved, the C2 is stable and it's able to express C1, which acts as a repressor, and it keeps off those early lytic genes, right? That's going to block the lytic cycle. In the other case with well-fed bacteria, C2 is broken down. We end up with an absence of C1, right? It's not expressed because C2's been broken down. And in this case, because there's no C1 to block this transcription of those phage DNA or the phage genes, we enter the lytic phase. All right. So here's a little more information about this. So basically, excuse me, uh, starved bacteria. So if there isn't much food around for the bacterial cells, and if there's multiple lambda infections, so if one cell is has multiple lambda bacteriophages attaching to it and trying to infect it, these things are telling us that or are letting the bacteriophage know, I guess, they act as a signal that it's not a good time to enter the lytic phase. It's not a good time to reproduce, right? So if bacteria is starved, it means there's not a lot of food around. There's not probably going to be a lot of bacterial hosts available. If it makes a lot of viruses, and multiple lambda phages trying to infect the same cell, that's also kind of a signal that there aren't a lot of hosts around, right? If they're trying to infect the same cell. And so these are conditions that aren't so good for the lytic cycle, for reproducing, making a lot of new bacteria or new viruses. So in that case, you end up with lots of the lambda CI, that's the repressor, you into the lysogenic cycle, right? The other side, well-fed bacteria, if there's just a single lambda infection, these are conditions where you end up uh, with the lambda CI not being expressed, or it's inhibited. And so there's no repressor to turn off those lytic genes. Those are expressed and we go through the lytic cycle. Now, these are environmental factors control the decision, and these can change, right? So the decision to go into the lysogenic cycle is reversible. And so something like UV radiation can affect and damage the DNA of the cell. It can cause the prophage to leave the bacterial chromosome. And in doing so, in that condition, we also end up with the lambda CI no longer being expressed, no longer being there as a repressor, and the cell can then, or the virus can then enter the lytic cycle.